Bismillahirrahmanirrahim. Assalamu alaikum class. In the previous lectures, we are talking about the penicillins. We have started the individual compounds or the different semi-synthetic, synthetic penicillins or the derivatives of natural penicillins. In the case of aminopenicillin, we have got the amoxicillin, but one of the problem in the case of amoxicillin or ampicillin is the diarrhea, which is due to the effect of the compound on the intestinal flora. We have got another compound, that is clavulanic acid. That clavulanic acid is one of the strong inhibitor of the beta lactamases, but is a weak antibiotic compound. Now we have got one preparation. It is available as augmentin. It has got the ampicillin with clavulanic acid. Ampicillin or amoxicillin can also be linked with that clavulanic acid that will reduce the side effect, that is diarrhea. Other derivatives are also prepared, which are termed as acyl uridoderivatives. These include azrocillin, piperacillin. In azrocillin, mazrocillin, piperacillin, NH2 group is converted into urea, or we have got amino urido group, and hence termed as acyl urido penicillin derivative. Azrocillin is penicillin sensitive. What does it mean? Affected by the beta lactamase enzyme, but is broad spectrum. Parental penicillin. In these azrocillin, mazrocillin, piperacillin, the derivatives of ampicillin, the D side chain is converted to urea and has termed as acyl urido penicillins. In the previous lectures, we have identified the 1960 as a crucial year. In that year, we have observed the problem for the Pseudomonas aeruginosa. These Pseudomonas aeruginosa, also termed as opportunistic pathogens, they are already present in our body, but they are controlled by the body defensive mechanism, but becomes weakened by the use of chemotherapy or by some other means. That is one of the real problem in the hospitals, which leading to the septicemia. The victim are prone to the infections, especially the lung infections. Carpenicillin, the broad spectrum compound, but acid sensitive, hence can be taken parenterally. It is also stereospecific. Carpenicillin is a product of carpenicillin. We have got another strategy. We have added the probenecid, which enhances the activity, but that is not actually the activity, enhancement of the activity, but it is going to reduce the rate of excretion of penicillin. Hence, probenecid used in association with the different penicillins. Different penicillins. We have got few other compounds like thianamycin or thianamycin, which was discovered in 76, used as a potent broad spectrum against Pseudomonas aeruginosa, but also acid sensitive. The difference in the other penicillins with thiamycin is that it does not have acyl amide side chain. There is another compound, olivinic acid, 100 times or can say 1000 times more potent than clavulanic acid, but it is nephrotoxic. Nocardiene and other compounds obtained from Japan soil, active against gram-negative as compared to gram-positive, but it has not got a fused beta ring system and hence also have got a different type of the mode of action, but is still studied the penicillins are termed as cell wall inhibitors. This is due to the mode of action, or this is due to mechanism of action of these beta-lactam antibiotics. They are selective, irreversible inhibitors of the enzyme transpeptidase. This transpeptidase is required for the synthesis of peptidoglycan. Peptidoglycans is a spongy cell-like substance. It is composed of a series of alternating moieties, that is N-acetyl glucosamine and N-acetyl muramic acid. They are the repeating units. They form a long chain or a polymer. The sugars are linked with peptides, and the cross-linking will provide a rigidity to the cell wall. Rigidity to the cell wall. This peptidoglycan cell wall is required for growth and development of the bacteria. The penicillins, cephalosporins, or beta-lactam antibiotics are going to bind with the penicillin binding proteins that may be called as receptors. At least we have got 7 types of the proteins. They are from PBP1 to PBP7, and different compounds are going to bind with the different segments or different proteins, and hence they have got somewhat different effects, but overall they are all selective, irreversible inhibitors of the enzyme transpeptidase. If we consider the different protein binding sites, for example, PBP1A and PBP1B are the transpeptide required for peptidoglycan synthesis, surrounded, and they are used or they are required for the cell elongation. PBP2 involved in maintaining the rod shape of bacillus. Inhibition result in ovoid or round forms that undergo delayed lysis. PBP3 required for septum formation in cell division. Inhibition results in the formation of filaments containing rod-shaped units that cannot be separated, not going to form the septum. PBP4 to 6 are carboxypeptidase responsible for hydrolysis of D-alanine, terminal peptide bond. Cross-linkage of this bond is necessary. Inhibition of the peptide cross-linkage or loss of D-alanine or bacterial D-alanine carboxypeptidase is observed as a result of these antibiotics. They are going to acylate transpeptidase and inhibit the role of these in the synthesis of cell wall, in the synthesis of cell wall.